What is cell cycle? Cell cycle is the series of events that lead to the division and duplication of a cell, producing two daughter cells. It is divided into two major phases: interphase, where the cell grows and DNA is replicated, and M phase, where mitosis and cytokinesis occur. Interphase has three phases: G1 or gap1 phase, S or synthesis phase, and G2 or gap2 phase. Cells that do not divide enter into G0 phase. Cells are able to enter reversible, quiescent, or irreversible, senescent, and terminally differentiated cells G0 state. Quiescent state cells remain metabolically active but no longer proliferate unless called on to do so. Senescent cells are dysfunctional cells that have ceased proliferation and are permanently withdrawn from cell cycle. Terminally differentiated cells have irreversibly lost its ability to proliferate. In cell cycle control system, cyclin act as regulatory component. Cyclin-dependent protein kinase, CDK, is catalytic and protein kinase regulated by cyclins. There are four classes of cyclin, each defined by the stage of the cell cycle at which they bind CDK and function. They are G1 cyclins, G1S cyclins, S cyclins, M cyclins. CDK catalyzes phosphorylation of specific serine and threonine residues of specific target proteins as determined by cyclins. Phosphorylation is transient and reversible. Once cyclin CDK complex disappear, phosphorylated protein dephosphorylates via phosphatase. Cyclin CDK's forms are G1 CDK, G1S CDK, S CDK, and M CDK. Cyclin D CDK4 and CDK6 found in G1 phase. Cyclin E CDK2 is found in G1S phase. Cyclin A CDK2 is found in S phase. Cyclin A CDK1 is found in G2 phase. Cyclin B CDK1 is found in M phase. Regulation of activation of cyclin CDK complex. Cyclins are synthesized and degraded each cell cycle, but CDKs are constant. Cyclin CDKs complex are inactivated by regulated proteolysis of cyclin. Cyclin destruction is ubiquitin-dependent proteolysis catalyzed by ubiquitin ligase, enzyme SCF, SKP1, culin and F-box protein, is responsible for ubiquitination and destruction of G1 cyclins. APCC or anaphase promoting complex cyclosome for S and M phase cyclins. Other mechanism to regulate CDKS includes phosphorylation dephosphorylation reaction and binding of CDK inhibitor proteins, CKIs. Phosphorylation dephosphorylation reaction. In yeast, it happens at tyrosine 15. In higher vertebrates, inhibitory phosphorylation at threonine and 14 by protein W1, inhibitory phosphate is removed by protein phosphatase, CDC25. Activation of CDKS is done at threonine 161 by phosphorylation done by CDK activating kinase. Binding of CDK inhibitor proteins have two types: CDK interacting protein/kinase inhibitory protein, CIP/KIP, family and inhibitors of CDK4 and K4. CIP/KIP are P21 and P57, and K4 are P16, P15, P18, and P19. Control of DNA duplication in S phase. SCDK activation at late G1 phase is responsible for formation of multiprotein pre-initiation complexes for DNA unwinding and replication. Origin recognition complex ORC. Mitosis promoting factor is synthesized in S and G2 phase. MCDK induce chromosome condensation, breakdown of nuclear envelope, assembly of mitotic spindle apparatus and alignment of chromosome at metaphase plate. Cell cycle checkpoints is to ensure that incomplete or damaged chromosome does not pass on to daughter cells. It is classified into cell cycle checkpoints, DNA damage checkpoints and spindle assembly checkpoint. Cell cycle checkpoint ensure fidelity of cell division. Checkpoint at G1 is called restriction point. At this checkpoint, cell becomes committed to DNA replication and completes a cell cycle. Spindle assembly checkpoint or mitotic checkpoint ensure that chromosomes are properly attached to microtubules and aligned at metaphase plate, prevents segregation of sister chromatids until they are properly aligned to prevent aneuploidy. DNA damage checkpoint is signal transduction pathway induced by damaged DNA. It is classified into G1S checkpoint, intra-S phase checkpoint and G2M checkpoint. Role of RB protein, pocket family. Retinoblastoma 1 gene encode RB protein. It is a nuclear protein 110 keta located at chromosome 13q14.2. Two other members are RBL1 and RBL2 for protein P107 and P130 respectively. They are tumor suppressor. The active or hypophosphorylation form of RB protein binds to transcription factor E2F and represses gene transcription, which is required for transition from G1 to S phase of the cycle. Phosphorylation of RB protein is initiated by cyclin D-CDK4/6 and hyperphosphorylated by cyclin E-CDK2 release E2F transcription factor. Role of P53 protein encode by TP53 gene. Its molecular mass is 53 keta. It is tumor suppressor protein located in chromosome 17p13.1 and it has 393 AA residues. In normal cell, it is inactivated by MDM2 mouse double-minute 2, upon DNA damage or other stresses. Various pathways lead to dissociation of MDM2 and P53. I response to double-stranded DNA, protein kinase ATM is activated, which in turn activates CHK2. It alters the MDM2 binding domain of P53. Hence P53 initiate transcription of P21, which acts as CKI, binds to G1 SCDK and SCDK to inactivate them. The another pathway is through ATR, hence, cell cycle will arrested to allow either repair and survival or apoptosis to discard damaged cells.